PET imaging, or positron emission tomography, is done to quantify the amount of Alzheimer's disease cytotoxic proteins in the brain. During a PET scan, specific radioactive tracers are injected intravenously into the patient. The radioactive tracer then travels through the vascular system and binds to a specific target in the brain. For example, AV1451, a tau tracer, binds to neurofibrillary tangles in the brain, which are aggregations of the hyperphosphorylated tau protein in neuronal cell bodies. The AV1451 binding to tau allows the PET scanner to pick up the radioactive tracer's location and intensity in the brain. Once the acquisition is completed, the PET scan is then reconstructed. It is essentially a contour plot where the red region denotes the high binding affinity and the blue region denotes low binding affinity. The problem is that the current generation of tau tracers are not tau specific. They not only bind to neurofibrillary tangles, but also other substances such as iron molecules and different tissue types in the brain. For example, here's a PET scan that has two different diagnosis results from two different physicians. One clinician determined that this person was tau positive due to the BRAAK signals at the arrow. Another researcher determined that this signal was just noise, since the person did not show any tau deposits in earlier BRAAK stages. Therefore, there's a need in the tau PET radiology to improve the inter-rater reliability of the tau PET images. So here is one of our initial prototypes that removes some of the non-significant PET signals. The image on the left is the pre-processed PET image, and the image on the right is the post-processed PET image. So in total, there are about three types of off-target bindings: stochastic noise, unpredictable off-target binding, and predictable off-target binding. Stochastic noise results from data acquisition and reconstruction of these images. Predictable off-target binding happens when tracers bind to known locations in the brain, but not because of the tau presence. Known locations can be basal ganglia and choroid plexus. And unpredictable off-target binding is when tau tracers bind to other substances that can be anywhere in the brain. For example, tau tracer AV1451 binds to mineral structures including iron. If a patient has microbleeds, the tau tracer will likely to bind to hemoglobin around the region because iron is an essential component of the hemoglobin. So here's the pre-processed PET image overlaid on top of the patient's MRI. As you can see, these signals cover all over the MRI and is not really region specific. After feeding the PET images through our program, a lot of the non-significant PET signals were removed, making it much easier for the clinicians to interpret the PET image and improving the inter-rater reliability.